Thank you everybody for joining us. We are today at our last UAC webinar of this 2024, and we are having an external speaker today coming from the University of Gothenburg, or rather he is at the University of Gothenburg, so he's joining from Gothenburg. In the University of Gothenburg, there is also a center similar to ours, the Center for Antimicrobial Resistance Research as well, and they have a good array of people working with different topics related to antibiotic and antimicrobial resistance. And today, Morten Grötli is going to talk to us about something a little bit adjacent, so not directly antibiotics, but other type of medchem project, which is developing of treatment for malaria parasite. And he's gonna talk to us about the advances in selective NMT inhibitors in a multistage approach to target Plasmodium, which is the parasite that gives malaria. So Morten, it's my pleasure to have you in our seminar series and ready to learn a lot with you. Like other times, this webinar is going to be a presentation first and then a Q&A at the end. And at the Q&A, you can either raise your hand and pose the questions directly to Morten, or if questions come up and you don't want to pose them yourself, you can write them on the chat and I will read them at the end during the Q&A as well. So with that, Morten, I give you the digital stage and ready to hear your presentation. Thank you so much for being with us.
Thanks a lot, and thank you for the invitation and giving the opportunity to present this. So you see the title here. It's all going to be about an enzyme called, or abbreviated to NMT, and how that can be used to potentially treat malaria. And we're talking about a specific type of malaria, which I will get back to. If we start just saying, okay, malaria, where do we find it? And if you look at this slide, you will see the yellow areas is where it's reported that you have malaria. And the interesting thing here is that if you look on our side of the map, you see here is Europe. Actually, it doesn't mean that malaria is a big problem in Europe, but several European countries report accidents of malaria or incidents of malaria each year. So it's spreading, probably because of the climate change. There is usually 5 types of malaria we talk about that can give diseases for human beings. And the most common one is marked in blue here, the falciparum, which you mainly find in Africa. But the one we're going to talk about today or focus on is the one showed in yellow. And we're focusing on the one called P. vivax. There are 2 other ones that also is covered by the yellow color. But vivax is the interest of, or the focus of this talk. If we talk about numbers, malaria, we estimate, or the current estimate is that around 600 to 650,000 dies of malaria each year. And we have something like 250 million people who's infected with malaria. This slide might look a little bit complicated. It just shows the life cycle of the malaria. And there's a few things I want to point out with it, so we're going to go through that. If we start over on the left end, the way this disease is transmitted to human beings is a mosquito that bites you, it sucks blood from you, and it brings with its sporocytes. These sporocytes, they will travel through your blood and to the liver, which is this gray part here. They infect the hepatocytes. They start to replicate, and they form what is called schizont, which will leave the liver again, get out in the bloodstream, and the blood stage is shown over here. It replicates again. It creates parasite or gametocytes, which is of male and female type. And these are in the blood. And when a new mosquito bites you, it will pick up this, and the whole circle is completed. Within the mosquito, these are developed even further and eventually become sporocytes again. and then we can repeat the whole cycle. Now, if you look at the lower end here, there are listed some drugs. And when it comes to what is called hypnocytes, which is this stage here, is actually a liver stage, but it's a latent stage. It means that the parasite just live in the liver for a while. For a while, it can be days, it could be weeks, it can be even months. And there are basically two drugs for treating that, these two. We have several other drugs that can stop it on the liver stage when it's replicating. And most anti-malaria drugs we have today, and most of the stuff that is in clinical trials, go for a blood stage. So we have many. And then finally, the gammocytes, there is only basically one drug that is used for that. Now, I want to go back to this dormant stage, because one of the big challenges with irradiating malaria is that normal treatment with drugs could be that when you measure the number of parasites in the blood, you can't detect anything. So you think the patient is healthy again. This is only now relevant for P-vivax. They are the only one that has this dormant stage. But then, even if you can't measure any parasites in the blood, it can take a few weeks, few months, and then the patient have a relapse. And it's because these dormant hypnocytes decide that they're going to go through this stage, they're released in the blood, and we start the whole circle again. And they think as much as 80% of the relapse of P-vivax malaria is due to these hypnocytes. But on the list here, it says that we have two drugs for these. And I'll point out a few drawbacks with these, which basically explain the reason for this talk or the reason for this work we're doing. If you look at these slides, this is a description or target description of what sort of properties drugs should have in order to treat different stages of malaria. And I put a red box for that one that is relevant for this story. And you see it's called anti-relapse. And this is just what I explained on the previous slide. When you have this dormant stage of the P-vivax, it can decide that it wants to go out to the liver stage again and start to replicate, and we get a relapse. So this TCP3 is a description of the ideal drug for treating this kind of malaria and prevent anti-relapse. And if you look at that, how that's defined, and this is done by the World Health Organization, you see that it's a minimum essential things, and it's how it should be ideally. But then an interesting thing pops up, and this is what I put in the box here. It says that it's an abbreviation. I'll explain what that is in a second. But it means that if you have a certain mutant, you cannot use the current drugs we have today. And so I listed two drugs. They work pretty well for quite a few patients. But in the area where we have P-vivax, the estimate is that as many as 35% of the population has this mutation. And when you have this mutation, these drugs becomes toxic to you and is not useful for treating malaria. The mutant I talk about is shown here. It's an enzyme that takes glucose 6-phosphate and transfer it to 6-phosphogluconate. And if you look at how this enzyme is coupled to this cycle here with NADP and NADPH, you see that you have glutathione that is on the oxidate form, and it's reduced to this GSH form. And that GHH form, that's actually a molecule that is used to mop up reactive oxygen species. Now, when you have a deficiency in this enzyme, your cells become much more sensitive to what is called hemolysis. It means that your blood cell just burst, they're destroyed. And if you have that and take primaquine, which actually oxidizes GSH to GSSG, it basically means you will have less glutathione to catch up reactive oxygen species, and it promotes hemolysis. That's why it's toxic. And basically that means that this 35% of the population that has this mutation, they can't use the current drugs that are used to treat this specific type of malaria, P. vivax, and in the liver stage, where it's just latent. So what could we do with that then? Well, we were definitely not the one that came up with this idea, but there have been people that worked on an enzyme called N-Myristoyltransferase. And basically what it do is shown on this cartoon here. You have ribosomal synthesis of protein. That's the red line here. At the end, you have a G residue. This enzyme, mucoenzyme A, what it does, it puts its fatty acid onto the G. And when this protein is folded properly, translated properly, it has this fatty acid on it, and that's essential for its biological function. It could be things like where in the cell it's going to be positioned, for instance, on the inside of the cell membrane. So this works as an anchor. It has to do with stability of the protein. It also has to do with interaction with other proteins. And it's been shown quite clearly that NMT is actually a quite good target for making antiparasitic drugs. But then we have a challenge. And I try to illustrate that here. Humans have two types of NMTs, these enzymes. And here, the malaria parasite has only one, and more specifically, the P. vivax have one. What is shown here is actually the mechanism for how this enzyme do its reaction, coupling the fatty acid to the protein. But the point of showing this is basically, if you look at the colorful spheres, that's amino acids in the active sites. And if you look carefully, you will see that this looks very similar, meaning that the human NMT and the P. vivax NMT has a very high homology. The homology totally is around 60%, but in the active site, it's more or less 100%. And then we get the issue, how do we obtain selectivity for the parasitic target over the human one? And if you think a little bit about this, if we're going to treat malaria patients with this becomes a critical issue. If we shut down the human NMT, it's probably not good because it plays a similar role in human cells. So it's very important for the function of a whole range of different proteins. So that will for sure result in or serious side effects. When we started this, we looked at some compounds that was already out there. And two of these are shown here, this EMP 1002 and this DDD 85646. Both these compounds have been developed by the Tate Group at Imperial College in the UK, who's done some really fantastic work on making NMT inhibitors targeting malaria for at least the last 10 years. And what is typical for quite a few of the compounds they come up with is that they're pretty potent. I just listed the IC50 values. Several of them are really low nanomolar. But the drawback is the following. If you look at the difference between the human enzyme called HS and the P. vivax called PV, you see it's very little. The selectivity index is around 2. And for some of the other ones, the selectivity index is, it's not 1 even. It means that the compound binds preferably to the human counterpart and not to the Vivax, or it has a preference for a human one. This picture that NMT inhibitors have low selectivity but high potency is also true for other parasitic targets that people have developed NMT inhibitors for. So the current state of the field is yes, we can make potent compounds, but the selectivity is far from optimal. Now, when we started to think about this, we said that why don't we look at how these inhibitors bind and see if we can figure out which interactions trigger any kind of selectivity? Could that be a possible way forward? That's basically the question we had. If you look at the color code now, this square here, it means the central core. The orange-yellow color means the head, and the blue color is what we're going to call the tail third row now. If you take these two compounds, put them into the crystal structure of the P vivax, you see that they bind in the same area, but they have different binding poses. The orange molecule, which is the 1002, shown to the far left here, is shown in orange here. You see the upper part here where we had the head, we actually have this amino function. And then at the other end over here, we have the tail part. And then you can consider this to be the sort of central scaffold part. Now, based on this, we actually thought like, okay, let's take a central scaffold that can position different head groups and tail groups in different ways, and then see what happens when we start to play around with this to understand a bit more about which interactions cause selectivity. And this just summarized the compound we're trying to use. Two different tail groups. We have a core A and a core B. The lower three is the core B. The upper part is the core A, and two different types of head groups. And again, I just showed here one of the compounds we made called 12E, where the blue box here indicate the tail region, and the upper part here is the head region. And if we look a little bit how we imagine making this, this is the retrosynthetic analysis of this class of compounds. As you saw, we had a B core that could be a monocyclic or bicyclic. It could have a heteroatom in here. We should have the head group, and we should have the tail. And then the idea here was that this kind of compound like this is commercially available. We could couple that together with a compound like this that will be the B core. So that will be palladium chemistry and some protection group chemistry. We could introduce the tail group onto this phenol, for instance, with a sulfonyl transfer or a Mitsunova reaction. We could make the alcohols that we have up here in the tail group from the ester show down here. So that would be a reduction. And to make the sort of core, the heterocycle in the tail reactions. And you see that we could then start everything out from a substitution reaction. You see the majority of the reagents we would need for this would basically be commercially available. So that seems like a decent way to do it. So I'm just going to show you how we made this. We're not going to dive into too many details, but just to get an overview of the chemistry involved in this. If you look in the corner now, I just showed the structure that we're looking at. And you see, again, the color code, so you can follow. If you see the line, the upper pathway here, it's basically to do the substitution reaction followed by a condensation reaction to get the heterocyclic core, and then do the reduction reaction. We could also introduce the tail with this sulfonyl transfer, showed here. We use the phenol, put on a leaving group, react with an alcohol, elevated temperature, and you introduce the tail. We can also introduce the tail by a Mitsunova reaction. Then we use the core that we build up, or the alcohols, couple it to the phenol by the Mitsunova, and you see in general that turn out to work better than actually doing the substitution reaction. We could then do the palladium-mediated Suzuki cross-coupling to generate these kind of intermediates in pretty good yield. And then what is left is actually to knock off the protection group. And then we will get the first zero compounds, which we here called 12. And you see it's in reasonable yield. Some things worked really well. Some things didn't work that well. I would say that the lower yields was probably mainly due to purification issues and handling issues, solubility, stuff like that, and not so much with the chemical transformation itself. We could also do the scaffold slightly different to get to the series 2 compounds. If you compare them, it's basically the head group that would be the different. In series 1, we have this cyclic moiety with an amino function. And in the series 2, we have a straight chain with an amino function at the end. Otherwise, they're very similar. And again, to do this, it's basically a palladium-mediated coupling reaction, Suzuki, and then the introduction of the tail by Mitsunov reaction. Again, everything from decent to quite good yield. Main problem was, again, purifications and not to the same extent the chemical transformation. Series 3 is drawn up here. And a way to obtain that is starting this 2,6-dichloropyrimidine microwave reaction, Suzuki cross for the Suzuki cross coupling, followed by a Mitsunov reaction. And then we get this intermediate, and then treatment with acid to knock off the Bock group, and we get this series 3 compounds. So it follows the same type of chemistry as I already showed you. The next sort of series compounds has a bicyclic B core. It's illustrated down here. And that we make from this bicyclic phenol, introduce the tail first by Mitsunov reaction. We can do palladium-mediated coupling reaction. We can then treat it with acid to cleave off the Bock, and we got series 4 containing the cyclic head group. If you wanted to have the straight chain with an amino function at the end, it's follow the path down here. It's first a Suzuki coupling followed by a Mitsunov reaction to generate this kind of compounds. So then you've seen the chemistry that was used for making these series of compounds. So did they have any effect then? Or what sort of effect did they have? Well, the first thing was to test them in a biochemical assay. And I'm not showing you the data for all the compounds, but just a summary of maybe the most important findings. You see the table here. And if you look at the first row here, you see the IC50 that was obtained in the biochemical assay for targeting the NMT from the PVVAX. And I would say that, yeah, it's pretty decent. There are several compounds that are in the lower part of nanomolar, but we also have quite a few that is, in the ballpark 10 to 30 nanomolar. So they're not more potent than what's been reported before. But the interesting thing is this row here. Remember, I told you that some of the compounds we started out with that was not developed by us, they are really low nanomolar inhibitors, so potency is excellent. But the selectivity was not very good at all. But look at this. We have compounds here that has a selectivity index. That means a preference for a PVVAX or immune ones. Everything from around 7.8 all the way up to 260-fold difference in selectivity. It's pretty good. And if we look at the compound now, again, I just summarized how they look. This is what you saw the synthesis for. If you look down here, you look some of the compounds that have this bicyclic core. And for the rest of the discussion now, there will be a certain focus on this compound 13A and the compound 12B. because they showed the best selectivity index. If you look a little bit on the structure of this, it might give us a hint why things are the way they are. If you look over on the A panel here, you will see that the myristoyl, the coenzyme myristoyl, is shown in green. That's that area over here. And what's showed in yellow here is one of the compounds, 12B, that had a very good selectivity index. And what you see, if you look at the global structure, the dark black line is actually the enzyme without an inhibitor bound in here. And the one in the blue turquoise color is the one with a compound bound. And you will see that some areas there is a very good overlap between the black line and the turquoise line, but other places there seem to be quite a bit of differences, basically telling us that when the inhibitor bind, we see changes in the whole structure. And if we focus in on the binding site itself, where 12B bind, what you see in this picture here is that we taken the complex with 12B, and if you look, the amino acid side chains that are marked in the turquoise color, that's from that complex. All the things that are in gray or whitish sort of color, that's from other complexes that we haven't made. And the green ones are from the human enzyme, the so-called NMT1. Remember, I told you there were two enzymes. We used NMT1 to compare with here. And what you should notice then is that you see, if you look at these complexes from the human ones, you will see that this amino acid here, this tyrosine, it's moving around. It's quite a bit of flexibility in it. While if you look at the PVVX complexes, the majority of them is pointing out in this direction, just like it do for our complex. What that tells us is that the plasticity or the movement of this amino acid is much bigger in the human enzyme than in the parasitic one. And if you look carefully now, you will see that there are several dark arrows. And that indicates the sort of movement we have of these side chains when they're upon binding. And you will probably see that the green ones, the human ones, seem to tend to move quite a bit, especially the tyrosine here. Here it seems also like that this one, the phenylalanine, that you find in both the human and the vivax, move quite a bit in both species, and so on. But what this basically tells us is that the moving of these amino acid has to somehow be involved in generating the selectivity for the compound. Remember, I initially said that the amino acid sequence in the active binding site is more or less identical for the two enzymes. If you look at a few other compounds too and compare it to 12B, you will see that over here we have an overlay over these three structures. This is one that we synthesized, this one on the left-hand side and the right-hand side is made by the Tate group. And again, if you overlay them and look at how they bind, you see that they cover basically the same space. You see the head group up here, and you see the tail group down here. But you see that the DDD 85646 shown in the violet color binds very different. The sort of central core is going in a completely different way than the other two compounds. And the other thing is that 12B and EMP 1002, they overlap very nicely in part of the structure. But if you look up here, you see that this other aromatic ring has not a perfect overlay, and also the end group up here does not overlap that well, which again hints towards what might give us the selectivity. If we focus in a little bit more, you also see two figures, the C and the D panel here. And it's also shown or illustrates some important things like the tyrosine residue here can either point towards us or be hidden more in the background. This is the showed in black here. And you see that the yellow compound again is 12B, while the violet compound is this DDD 85646. The other thing you should notice is that although both have a similar head group, it's only our compound 12B that seem to make these key interaction with the neighboring amino acid side chains, while this compound, the DDD 85646, does not position the nitrogen in the right position and is therefore lacking these interactions. Again, pointing out that these head group interaction could be really important for selectivity. Let's see, I just have to close a window here. If we then look at the D panel here, you also see that there is a dotted line to a single water molecule. And it seems like that water molecule is also important for gaining selectivity. You see it's making a hydrogen bond with this nitrogen sitting in the yellow molecule, which is 12B, while the orange molecule does not have a functional group that is positioned in such a way that it can make this interaction with the water. Oh, it looks like my system is hanging up here. Just a second. Okay. If we then look at the toxicity of these molecules, we can first look at blood cells. And if you look here in the A panel, we just have an increasing concentration of the inhibitor. And then we see how the cell reacts to that. And you see that when you start to crank up the concentration, we start to kill cells. But we have to get up to well above 10 micromolar, except for the red line here, to really see a significant impact on the viability of the cells. If you look at the B panel, we have tested them on the blood stage development. And you see that by increasing concentration, we killing parasites. You see the different derivatives. Here is only showed the one in the 12 series. If we go up here, it's some other compounds in the 26 series. They also have a similar trend. Increasing concentration kill the parasite. And the last one is this with the bicyclic core, showing also that they kill the parasite by increasing concentration. If we then look at how is it at the liver stage? Remember now I told you that we had two types of liver stage parasites. One, the schistons, that basically is in there just for a short while to replicate. And we had the other type, where it's more a latent form that is just hanging around the liver. And again, if you look in the bar graphs up here, you see how the compounds do that. This is done at 20 micromolar. And you see that in general, it kills the parasite quite efficiently. It means that more or less all the compounds show decent to quite good activity, and some of them is quite potent and seem to kill more or less all the parasites. If you divide that up to the groups, that's shown in more detail this in the lower panel, the C and the D panel. But it's basically just another representation of what you see in the bar graph up here. In all cases, it seems like the compounds are quite active, especially between 10 and 20 micromolar. However, what is the really interesting thing is that the compounds seem to be very efficient treating for hypnocytes. That's the Latin form of the liver stage. And again, if you look at the bar graph up here, you see that some of the compounds basically wipe out all the parasites. It kills them very efficiently. Again, this is at 20 micromolar concentration. Again, this is, if you look here in more detail on it, you might say, yeah, Morten, that looks okay, but look at the error margins you have down here. Now, here comes a very important thing to remember then. When you want to work with these parasites, be aware that P. vivax cannot be cultivated in a lab. So the only way to do this test is that you collect blood from patients that have P. vivax infection. Then you let mosquitoes drink from that and infect animal systems. And then you have to take out the liver to measure how efficient you kill the parasites. So it's a rather complicated thing. And you get usually quite big variations in the data set when you have to do it this way. But unfortunately, there is no other way to do this. If we then look at how we can summarize then this, in the panel over here, you see some of the compounds, the types of compounds, the 12 and 16 series, the 26 series, and the 30 series. I try to illustrate the different types of compounds. And if you go to the B panel now, at the x-axis, you have the potency in nanomolar, and at the y-axis, you have the selectivity index. And you see that we have quite a bit of compounds that have a potency or an IC50 in the ballpark, 50 nanomolar plus minus, and they have a selectivity index from the one that's outlined here, above 100 up to 260, which was the best one. And if you look at this plot, it's slightly different and just say, what about the Plasmodium inhibition in general, where you have the blood stage IC50 value up on the y-axis and the hypnozoites at the x-axis. You see also that we have some compounds that are quite decent. And what this basically tells us is that NMT inhibitors, if you can get them selective enough, might be a very good way to have multistage inhibition. And then, if you summarize the data I showed you, you'll see that 12B and 30A is the compounds that seem to have the best properties. They're quite efficient. Now, based on this, we also asked the question, okay, can we do something else? Because what I showed you now was done a little bit on trial and error. And now we asked the question, what happens if we start to play around with the tail group, indicated by the blue dotted circle over here? And again, what this comes from is that if we move over to the A panel, you see our compound 12B, which I just showed you. And if you look down here, we have another compound that collaborators had identified through a high throughput screening campaign. And what is interesting with that compound is that it has an extension in the tail area, this morpholin ring here. And then the question was, since this can bind, it's possible to extend the tail. Could we gain more selectivity and maybe also potency by merging or making hybrids between these two molecules? And that's basically what we try to illustrate here. If you put them on top of each other, you see what sort of space they fill out. And we see that, the tail part is over here. And if you look how that looks in the crystal structure, if you dock them in, that will basically be to have compound 12B with an extension down here. And then we thought, okay, to do this, we would like to make a series of compounds that showed in the E panel here. It's going to be a head group. We're going to have a central scaffold, and we have the tail and tail extension group. And we thought that the A group, this core here, should then either be a pyridine moiety like this or this bicyclic moiety in the series 10. And the tail group extensions was based on doing careful docking, designing molecules that we think can make interesting interactions and make them. And we picked out this small series of compounds here. Again, radiosynthetic analysis to see how we can make them. This is the general structure. We think that we could divide it between the pyridine ring and the core molecule. That would be palladium chemistry. We could introduce the tail part onto something like a carboxylic acid moiety. We could probably take the core A, again, use the same technique as I showed you, to use a Mitz-Novo reaction to actually introduce the tail part. The chain here that generates alcohol could come from an acid catalyzed hydrolysis of a vinyl ether. And to introduce this chain, we could probably do palladium chemistry. To get the halogen in could be a bromination. And to make the heterocyclic tail part would be a condensation reaction. And then again, if we look at how we actually did it, condensation reaction to make the heterocyclic scaffolds work pretty decent. NBS to introduce the halogen, a Stiller coupling to actually introduce the vinyl group. And then we had this idea that we could do a two-step procedure to get the alcohol. And you see that, yeah, it worked, but only 20% yield. It was tricky to get the reaction running properly. And therefore, we ended up doing this procedure instead. It starts out the same way, condensation reaction to actually create the heterocyclic scaffold, bromination by NBS. Then we use a palladium-edited Suzuki coupling to introduce the precursor that's going to be the alcohol. We hydrolyze that to get the aldehyde and then reduce it down to the alcohol. And now you see the yields are much better. It's a little bit more synthetic step, but it works very nice. And then to make the series 1 and series 2 compounds, that basically is with a different sort of core A scaffolds. It's outlined on this slide. Starts from the starting material to the far left side, doing a Mitz-Novo reaction. And you see the yields are quite decent. We then hydrolyze these esters and do a coupling to the carboxylic acid. So it's an amide coupling. Then we have introduced the side chains here. And then we follow up with doing a Suzuki coupling, followed by acid hydrolysis to remove the Bock group. And we have the series of compounds. And they were all obtained in what I will call good to excellent yields. If we then go and look at the biochemical assay, what sort of IC50 values did we get out of it? And you see here we tested both for the P vivax and for the human counterpart. For some of them, you see, we didn't measure for a human counterpart. And in those cases where the P vivax IC50 was, around 1,000, we said it's not interesting to compare to the human one because they're not, the P vivax activity is not good enough. That's why it's lacking, for instance, for this compound. But you also see that when we compare these two, we get some kind of selectivity, but it's not as good as we had in the previous types of compounds. It's everything from 1.2 and up to maybe 50, 60 in selectivity index. And if you look at the other one where we have extended the A core to a slightly larger group, so it's bicyclic, you see that we do the same measurements. We have one compound that has a selectivity index of 125. It's reasonable. And then we can ask, why is it like this then? Well, again, if you look in the A panel here, you see how the compound binds. This time we also illustrated that, there is some dots sticking up here, which is a water channel. And the myrticityl coenzyme A is binding over here. So the binding site you see for these compounds are the same binding site as we had for the previous series of compounds. That's nothing new. And again, if you look at the compounds now, you see that introducing the tail here, it looks pretty crowded. And this is basically based on two structures, number 9C and 10B. And one of these compounds does have the A core as a single aromatic moiety, and the 10B is this bicyclic moiety. But what you see here is that they seem to cover quite a bit of space here. The extensions are in here. And again, we see that this phenylalanine moves quite a bit, indicated by this arrow. So I mean, the selectivity obtained is again due to the flexibility of some of the amino acids. But we don't get the same selectivity indexes or as good selectivity indexes as we've seen in the past. I'll come back to explain why that is. If you go down to the C panel, you see that in the smaller box here, that's highlighted here, where we looked at the interactions between this single tyrosine and part of the molecule. And now we call this the inner wall because this is an amino acid that actually defines the binding site for these inhibitors. If you look at the bigger box here, it actually looks at what happens with this hairpin down here. And when you zoom in, you see that the binding of these different compounds, 9C and 10B, has quite a bit of impact on how this loop look. So again, there is quite a bit of flexibility, plasticity of the enzyme. Now, if you look in the upper part here, you see actually one compound, but it looks like several of them. And that's because when you look at the electron density for these compounds, it can give different interpretations. It means that it's not locked in one position, but you can have different interpretations and see that, it's not locked in one position. There seems to be no preference really, but it can cover a certain area. And if you look at the B panel here, it's just illustrated with two variants of the same. And that is that, although it's the same compound, the orange and the yellow does not overlap in the tail area at all. Or I shouldn't say at all. They cover roughly the same space, but they look quite different. There doesn't seem to be a clear preference. However, if you look at 10B, which has this bicyclic A core, defined, it's a morpholine ring sticking in here. It means that it's much less flexible. So we just see, the interpretation of the X-rays is basically two, maybe three different ways to position the morpholine ring, while these open chains can be positioned in many different ways. Basically, what that means is that this is more locked, it's more rigid for the morpholine ring, while these open chains are much more flexible and does not seem to be good for high potency and for good selectivity. I also said that we looked at how water molecules might play a role in this. And if you look at the A panel, all these dots up here are water molecules. This compound 1 is the one we had as 12B on some of the previous slide, which was one of our best hits. And you see that makes an interaction with a water molecule here, and it makes another interaction with a water molecule here. However, if you look at 9C, which is molecules that has this extend, or the molecule that has an extension in the tail area, and you see it has two different positions here. You see that it doesn't manage to make this, partly because it doesn't have the nitrogen here, but also the part over here does not position the nitrogen to make that tight interaction with this water molecule. If you look at the one with the bicyclic core, you see the morpholin ring is well defined down here, stuck in one position. We compare that now with 9C, the same as you have in green in the A panel. And you see that the tail here is in different conformations. You see that the nitrogen here can partly make the water interaction, but the bicyclic core does not contain anything can make that water interaction. You also see that it's something called inhibitor-free. It means water molecules are in more or less the same positions, even if you don't have an inhibitor here, meaning that the water molecules we're looking at here is actually locked in the crystal structure. If we look here, we actually look at the head group, and we have not done any changes to the head group. But if you look in the A panel, you see the tail is quite flexible, cover a large area. And then if you look carefully here, you see the nitrogen here at the head group, it makes interactions with some amino acid side chains and some water molecules. But the interesting thing now is that if you move that over to the A core that is bicyclic, you see that pattern change. This N161 does no longer make any interaction with the head group, although the head group for these two molecules are the same. So it's just because the core here has been changed to a bicyclic moiety, the interaction with the N61, that seems to be quite important here, it vanishes. And that's probably not a positive sign. And if we then look at this on a more globular way, if you focus now on panel B and panel C, this one, the panel B, shows the changes we have when the inhibitor bind to the vivax, either the 9C compound or the 10B. The 10B is the one with the bicyclic moiety. And again, the arrows, if you look at them, that might make it more easy. You see that again, the arrows, there are some quite big movement on certain amino acid. What that means is that, again, this plasticity or flexibility of the molecules is important for making selectivity. And it's a little bit the same for the human counterpart, but I can't draw the arrows here because now we have docked things in because we don't have a crystal structure of 9C and 10B in the enzyme. So these are dockings, while these in the B panel are crystal structures. And that's why you see much less movement of the phenylalanine, for instance, over here, compared to what you have in the PVVAX, because we have crystal structures for all these.
Hi, Morten. Just letting you know there's 5 minutes left.
Yes, I'm just to wrap it up.
Sure.
If we, again, just look at the B panel here, in the thin lines, you see the two inhibitors. And in the thicker line, you see the amino acids that move around, the differences between them. And then you see that if I have 10B in here and compare that to the NMT1, you see that the amino acids looks a little bit different. Again, reflecting on this, that the flexibility of this amino acid plays an important role. That's a highlight of what is showed in the box here, this area down here. Now, what did we get out of this then? Well, it means that compounds like 12B, it's bind selectively to the vivax, but it's all because of very specific interactions. And the selectivity is enhanced by structural plasticity. And I showed you several times that, for instance, tyrosine 2211, it can rotate quite a bit, but it's very different between the the P vivax, and the human counterpart. If you look at compound 9C and 10B, these were the one with the tail extension. They also endure major conformational changes, and these changes contribute to selectivity binding over the human counterpart. 30A and 12B are the most selected compounds we have, in the ballpark 260 to 270 fold selectivity index. And what this means is that what we call the cryptic pocket is basically where the inhibitors bind. There are certain amino acids that seem to be much more important for gaining the selectivity due to this plasticity. And we also learn from this that expanding the binding site with bulk extensions that seem to have limited efficacy compared to stabilizing specific conformations. And we also, I hope that I showed you that although the PV, NMT, and the human counterpart are very similar in amino acid sequence in the binding site, this flexibility actually opens up the possibility to make inhibitor design with some decent selectivity. And if you look at this in a broader sense, I would say that I showed you data showing that if you can get the selectivity, this might be useful for treating both blood stage, liver stage, including the dormant P vivax liver forms. And we don't really have any good drugs for doing that due to this mutation of certain enzyme that many people have. And this versatility of the NMT peptide binding cleft that offers opportunities for multistage anti-malaria drug design, meaning covering both liver stage and the blood stage. Just one little slide and say, so what's next up for us? Well, we think that based on the data we generated so far, we will prioritize conformational changes and target subsize to improve inhibitor selectivity. And we plan to do this now by using artificial intelligence run program. We're going to use something called Reinvent, which is a program that you feed in the data, and hopefully we hope to develop that in such a way that we can use it to tune the selectivity towards high selectivity for the P vivax or the human counterpart. These are the people who's been involved in this. Just like to point out a few things. All the chemistry, all the compounds is done by Diego, very talented postdoc that's been in my lab. The structural work is done by the next 5 individuals. And all the testing of the compounds is done by all the people over on the right-hand side here. Funding, I would like to point out that we used funds from these different funding agencies, but the project was more or less done without any funding that was specifically dedicated to this project. We leaned on other resources we have. And very much of the success of this project is due to Diego, who did a fantastic job. And that's it. Thanks a lot for listening. Happy to take questions.
Thank you so much, Morten. We don't have a lot of time for questions because it's already past and it was scheduled from 12:30 to 1:30. But if there is any questions, anyone has a question, they can put their hand up. We can at least have one. If not, meanwhile, I can ask you something that I've been thinking about. So my background has nothing of chemistry, so I'm not going to ask you anything about the chemistry. But I was thinking it's the first time that I hear a talk about drug development and design for a parasite that has enzymes that are very similar to the human body. Like you were explaining, this is the key thing that you were about the selectivity. So something that came to my mind as you were talking was the availability of these 2 different targets in the human. Because if I think about it, I would expect a human that is infected with Plasmodium. Both Plasmodium and our cells have this NMT enzyme, but there should be much more of the human one than of the Plasmodium one. Is this something that plays a role into how you work for the IC50s and the selectivity ratios, or how does the medchem goes about it when there might be more of one enzyme than the other that you're trying to select for?
Yeah, that's a good question. I mean, probably next year we will try to do the first animal studies for this. That will give us more a hint about how to do dosing to see the effect. Uh, I mean, we're far away from treating human beings with this,
Yeah. Mhm.
but let's say like this: if we have a mice that's been infected with a PV vax, right now we don't have very much clue about what sort of dosing is required to
Mhm.
mainly see the effect on the VVAX parasites. Uh, the other thing one should also mention is that the Tate group that did a lot of this work in the past but didn't get high enough selectivity, they actually started a company that specialized on treating cancer with NMT inhibitors, and it goes really, really well. And again, the thinking behind that is that certain cancer forms, not all, seem to have a much higher expression of NMT just because the cells are growing rapidly. And they
Mhm.
have some really nice preclinical data showing that some of these inhibitors that were originally developed for parasites is actually quite powerful ways to treat cancer. But what that implies for us is that they've done um, studies in animals already, and they don't seem to have severe toxic effects, which probably implies that, by playing around with doses, it might be possible to get around this issue. You just mentioned that maybe it's more NMT in the humans or in the
Mhm.
animal models than that, but we don't know that yet. So that would be exciting to see.
Okay, thank you so much. Um, Dharmet has a question. Dharmet, yes, please.
Yeah, thank you very much, Morten. Like Ava, I'm not a chemist, so I have a question on a different angle.
Okay.
I think it was very interesting, and you showed us wonderful detail on your chemistry plans and how they interact with the structures. So I'm thinking further down the road, how does this affect IP or the ability eventually to develop a drug that can be brought to patients?
Oh, that's also a very good question. I mean, the way we looked at it is the following. We would like to, focus on parasite diseases, and it's not really that much money to make out of that. However, when that is said, we've been in contact with the Malaria Venture and the World Health Organization to see if we can get some support to take this to the next stage. And basically, their recommendation was patent it. And they said, why do you want us to do that? We're not interested to make money. But they said, no. But if you have a patent, we can keep the industry, the pharma industry, away from this. So we would like you to have a patent on it. Then we're much more keen on helping you because then you decide what to do with it. Uh, the Malaria Venture has told us that in general, all sort of compounds that reach clinical trial and looks very promising get patents on it because then we can make sure the pharma industry doesn't jump on it. And then we run into these issues. So actually, we've given away the right for all of this. We said that our American collaborators that are much better set up to do clinical studies for this, we're giving it all to them. So all the work you've seen is published. It's also all patented already. And we will try to keep it that for the future too, just to make sure that it reach out to as many people as possible.
Okay, thank you. So it is protected because I think regardless of keeping big pharma away from it, if eventually you want to involve big pharma to provide the money to develop it, they will anyway want patents to protect themselves against other big pharmas.
Could very well be.
Yeah.
That's a complicated business.
Yeah.
True.
Anyway, thank you. Very exciting.
Oh, nice to hear. Thanks.
Yes, thank you so much. Thank you, Darmet, for your question. Thank you, Morten, for your time and preparing this presentation for our seminar series. It was great to have a different angle on MedChem in our work here. And I hope to see how this develops and what else comes from your lab, Morten. Thank you so much.
Thanks a lot. It was a
And
pleasure
thank
to be
you.
here.
Thank you, everybody that joins us. We will have a new webinar series for the upcoming year, 2025, with some exciting news because our PhD students are going to start presenting their projects and their the work that they've been doing over the past 2.5 years, all of them. So keep an eye on that coming out in January, February with our first webinar for 2025. Thank you, everybody, and have a wonderful, wonderful holidays time. Bye.
Bye-bye. Thank you.